Thank you very much, Isabel, and thank you to the organisers for the invitation. It's an honour to be presenting this morning on hepatitis C. It's a particular pleasure to be presenting at such an exciting time for hepatitis C with the rapid pace of recent therapeutic development. I'm going to give you an overview of recent therapeutic development and then move into issues of therapeutic access. I have professional relationships with several pharmaceutical companies, and I'll be presenting some data on unapproved medicines. The development of interferon-free HCV therapy provides one of the major advances in clinical medicine in recent decades, and it truly is a leap forward as far as clinical management, and I think it really underpins our advocacy towards interferon-free access to all. If you think about therapeutic development in terms of how well tolerated regimens are and how effective they are, we've come a very long way from standard interferon. The addition of ribavirin, the pegylation of interferon, and then the addition of the first generation protease inhibitors, telaprevir and boceprevir, all increase the efficacy for hepatitis C, but at a tolerability cost. It was only with the advent of the next generation of protease inhibitors, cimeprevir, and then the development of the nucleotide analogue sofosbuvir that we started to move to the right with both increased efficacy and improved tolerability. The great leap forward obviously has come with the removal of interferon and the development of several interferon-free regimens. It took us 20 years to get to the first generation of protease inhibitors, but it's only taken 3 years from that point to the development of highly effective interferon-free therapy. We still probably have a few years to go to get to perfectavir, a term coined by a colleague of mine, Jordan Feld, and I'll later in the talk go over the aspects of the so-called perfectavir solution. I just wanted to touch on some recent developments in interferon-free therapy. There's several highly effective interferon-free DAA regimens for genotype 1. There's also development for non-1, although it's a little behind the initial development for genotype 1. Obviously, for this audience and for myself as someone who's been involved in management of HIV, hepatitis C co-infection, it's particularly encouraging to see the outcomes within the context of co-infection, where there's no impact of HIV on hepatitis C treatment response. There's still some work to be done to characterise the impact that potential resistance may have in the context of clinical management, but I believe at present that HCV resistance testing is of limited clinical relevance. We thought that there may not be much individualisation at all recently, but I think there will remain some individualisation, at least for the coming years. That's particularly related to disease stage, where patients with cirrhosis may require longer duration of hepatitis C therapy. They may also require some genotype-specific individualisation until we get highly effective pan-genotypic regimens. One of the important clinical management questions is whether decompensated cirrhosis is reversible. I think the early studies are showing that it is reversible, but certainly not always. Even in patients that have clearance of the virus, many patients are too far along the sort of liver failure road, so to speak, to obtain significant reversibility, and they still progress to transplantation. Shorter duration therapies are certainly on the horizon, and with them come the potential for pan-genotypic potency with triple combination therapy, and I'll show you some preliminary results of those regimens. I'm not going to go through all these regimens in any significant detail at all, but I just wanted to highlight the fact that there are several classes: protease inhibitor, nucleotide analogue, non-nucleoside analogue, S5A inhibitor, moving through, and different combinations with agents from these different classes. The other point I wanted to make is that there are several pharmaceutical companies involved in therapeutic development in hepatitis C, and I think that pharmaceutical competition will be crucial for achieving lower prices in the future, and that will clearly be required to underpin improved access. Just to show you a few top-line results in terms of recent trials and recently approved and some as yet unapproved regimens. So the SVR12 is a sustained virological response rate at 12 weeks post-treatment, measured by undetectable Hep C RNA, and equates to a cure of infection. And it's clear from all these results that the efficacy is highly impressive, with response rates at or above 90% marks. And that's with sofosbuvir, ladipasvir, with the triple combination regimen, paritaprevir, ombidasvir, dasabavir, with ribavirin, another regimen with the triple combination, then regimen grazoprevir and elbasvir, which is a protease inhibitor with an NS5A inhibitor, and sofosbuvir and cimeprevir, which in fact was the first regimen that was used as an interferon-free regimen for genotype 1 following the licensing of the individual agents of sofosbuvir and cimeprevir. So moving to the results in co-infection, it's remarkable how similar these results are. So with all these leading regimens, there have been studies that have been undertaken in both the monoinfection population and the co-infection population, with results showing 95% to 97% cure rates in both populations. Clearly, there are some additional issues in the context of HIV co-infection. There are significant drug-drug interactions that have to be looked into with some of the regimens. An example of that is efavirenz, has a considerable drug interactions with the NS5A inhibitors, so it makes it a combination that is not to be used with sofosbuvir ladipasvir, with sofosbuvir decladisvir, another NS5A inhibitor. However, the dose can be increased because of that interaction. So there's specific clinical management issues that will need to be dealt with in the context of co-infection, but these results are really quite remarkable. And when we think back to the era of pegylated interferon ribavirin, where the clinical trials consistently showed SVR rates 10, even 20% lower than the monoinfection population, this is really an impressive step forward. I wanted to look a little bit about the issue of safety of these regimens. This is sofosbuvir and ladipasvir in randomised trials, comparing that regimen with and without ribavirin. If you look at two particular patient-reported outcome measures, the SF36 or the FACIT-F, in the green are the ribavirin-free arms. So this is interferon and ribavirin-free. In the blue are the ribavirin-containing arms. So you can see that even during therapy, so when people get to week 4, week 8 on treatment, they are seeing improvement in quality of life. So not only are they not getting toxicity of therapy, but they're getting improvement in quality of life. And I think that's telling us something about the effect of suppressing the hepatitis C virus quickly with these potent agents. And it's telling us that the virus itself is having a direct effect in terms of impairment in quality of life. Once you look beyond treatment, so 12 weeks beyond treatment, even the ribavirin-containing arms, you're getting, with clearance of the virus, sustained improvements in quality of life post-treatment. So this is not related to, obviously, liver disease progression, or regression. This is related to suppression of the virus. Just moving just for a short period to the issue of HCV resistance. This is some data from sofosbuvir-cimeprevir trial recently reported, looking at the Q80K mutation, which is a NS3 protease polymorphism, which is quite prevalent among patients with genotype 1A. In fact, in North America, about 40% of genotype 1A patients will carry the Q80K polymorphism. In the context of pre-cirrhosis, so those patients that have F0 to 3 on disease staging, there's no impact of the Q80K in terms of response. However, in those patients with more advanced disease with cirrhosis, it appears that the presence of the Q80K does compromise the treatment response. There's also some compromise in terms of NS5A baseline resistance to some regimens that needs to be further looked into. So I think the jury is still out a little bit in terms of the clinical impact of baseline pre-treatment resistance, but when you have regimens that are providing 95, 97% cure rates, obviously there's a minor impact, it's not a major impact. I think the resistance story will probably be more of an issue post-treatment to look at people with resistance post-treatment to try and construct a retreatment strategy. Just staying on the issue of people with advanced liver disease, this is a large randomised controlled trial as part of the phase 3 program that was done with paritaprevir, ombidasvir, dasabavir, looking at 12 versus 24 weeks in both treatment naive and treatment experienced patients. And although there was no significant statistical difference between the two arms, there was a numerically higher response rate in the 24-week arm. But 92% for 12 weeks is not a bad cure rate for people with very advanced liver disease. When they looked a bit more closely in terms of the subpopulations, this is the 12-week data, dividing the population into the treatment-naive group, the treatment-experienced patients who'd had a prior null response to pegylated interferon ribavirin, so they were unable to suppress the virus completely, or a partial response or a post-treatment relapse. And it was clear that those that had their treatment outcome compromised were those that were treatment experienced with a prior null response. When they were treated for 24 weeks, this did appear to improve their response rate. So this led to the label for this regimen stating that those patients with cirrhosis who were treatment experienced should be treated for 24 weeks. So I think there will be some individualisation in terms of duration of therapy, as I said, predominantly based on stage of disease. Just moving briefly on to the non-1 genotypes. So there are some encouraging preliminary results with some regimens in terms of genotype 2. We already know that sofosbuvir and ribavirin, which is approved, is highly effective when given for just 12 weeks with a response rate above 90%. We also have sofosbuvir and decladisvir, which has pan-genotypic activity, although with small numbers showing highly effective results. And sofosbuvir and GS5816, which is a second generation NS5A inhibitor, equally effective. With genotype 3, it's interesting that 12 weeks of sofosbuvir and ribavirin really has quite suboptimal response rates. Those responses can be improved with extension to 24 weeks, but the answer for genotype 3 will be combinations with NS5A inhibitors. So sofosbuvir and decladisvir and sofosbuvir and GS5816 showing response rates at the 90% or above mark. So that development to date is really pretty impressive, particularly over the last few years. But further development of interferon-free HCV therapy towards pefectavir, I believe, will provide the foundation for broadened access to treatment. So what are the key attributes of pefectavir? Clearly, we need regimens that are highly effective, that are well tolerated, preferably once daily dosing, that have pan-genotypic activity, and short duration would be helpful, particularly in some settings. But the key characteristic of pefectavir will be that it needs to be affordable. It needs to be affordable in high-income countries. It needs to be affordable in low and middle-income countries. Just looking at the pan-genotypic question, this is further data from sofosbuvir and GS5816, which clearly shows, although in smaller numbers in a phase 2 trial, efficacy across the genotype distribution. The phase 3 data from this combination should be reported later this year and is obviously eagerly awaited. How short can we go? This is some data that was presented earlier this year in Vienna, looking at that combination of sofosbuvir and GS5816 with the addition of a protease inhibitor, GS9857. So it's clear from this proof of concept study that 4 weeks of therapy for chronic hepatitis C is a bridge too far in terms of shortening duration. 6 weeks may be possible, particularly in the treatment-naive population. So there's obviously more work to be done in terms of how short we can go. A lot of clinicians are saying, well, why do we need to get down to 8 weeks? 12 weeks is not very long. But there may be some settings, and I'll talk later about the prison setting, in some prisons where there's a lot of transitioning in and out of prison, so the shorter you could get the duration of therapy in that setting would be preferable. And then there may be some other advantages to obviously short course therapy. But the elephant in the room, as everyone says, is the price of these new agents. With the licensing of sofosbuvir and cimeprevir, that certainly set a very high watermark for the pricing of an interferon-free combination at $150,000 US. The subsequent listing of sofosbuvir ledipasvir brought the price down to just over $90,000. With paritaprevir, ombidasvir, dasabavir, just over $80,000. We're not too sure what the Merck regimen will be listed at in terms of price. Hopefully it's a bit lower than the other two regimens. But it's important to state that there already is significant discounting that's happening in the United States in terms of payers negotiating considerable price reductions. So most of the large payers in the United States are paying somewhere between $45,000 and $50,000 per course of therapy. I think as I said at the outset, continued pharmaceutical company competition hopefully will continue this trend down in terms of pricing for high-income countries. This high prices of therapy is obviously led to public health prioritisation. So who do we treat with these very expensive therapies? This is some data that was put together by a group that I was involved in, led by Lynn Taylor in Rhode Island, where we looked at the restrictions on Medicaid access to sofosbuvir in the United States. And obviously there is some heterogeneity in terms of response, but most people are restricting to people with advanced fibrosis, so F3, F4. Some states it's F2 to F4, and some states it's only patients with cirrhosis having access to sofosbuvir-based therapy. And you can understand these restrictions in terms of the potential budget impact these high-priced regimens are having. So this is a pragmatic public health policy. What is different, though, is the restrictions based on illicit drug use. So in that same review, we looked at the states that had restrictions on access to sofosbuvir based on illicit drug use. And again, there was some diversity, but 34 of the states had specific restrictions on access related to illicit drug use. The period of abstinence did vary. Some states were even restricting people who inject drugs or had injected drugs who were on opioid substitution treatment but were not actively injecting. So you could be a patient with cirrhosis who was on methadone, but in some states you would not be able to access sofosbuvir therapy. This is despite the preliminary data, at least in patients on OST, showing that there are incredibly good outcomes with these new regimens in that setting. Clearly, we do need more data in populations of people that are on OST who are also injecting, populations of people who are injecting who are not on OST, and those trial results hopefully will come forward soon. There's a large study with grazoprevir, elbasvir, that enrolled 300 subjects on opioid substitution treatment, many of them who are still injecting, that will report the data later this year, and that's obviously eagerly awaited. But these restrictions go completely against guidelines that have been developed. So AASLD, IDSA, HCV treatment guidelines state that recent and active injecting drug use should not be seen as an absolute contraindication to HCV therapy, and that the scale up of HCV treatment in persons who inject drugs is necessary to positively impact the HCV epidemic in the United States and globally. So not only do they go against guidelines, they're unethical, they're a violation of human rights, and their legality should be challenged. So what are the strategies that we should look at in terms of drug price reform? Pharmaceutical industry competition, I've mentioned a couple of times. Discounting, I've also mentioned. Risk-sharing arrangements. Australia's entered into a negotiation with the pharmaceutical companies to have a risk-sharing arrangement that I'll talk about a little bit later. Volume taxation. There are voluntary licenses. Gilead has developed partnerships with generic companies in India to produce sofosbuvir-based regimens for 113 low and middle income countries. And although there are significant countries that are not contained within this agreement, it is a step forward in terms of improved access. Some countries will take the pathway of compulsory licenses. So I think it'll be a mix of voluntary licenses and compulsory licenses in the low and middle income country setting. And we need continued advocacy and activism, and that certainly drove the way forward in terms of access to antiretroviral therapy in the context of HIV, and I think that can equally drive forward the efforts for improved access for hepatitis C. Even if we had therapies that were very, very cheap tomorrow, it would not be the total solution. So there are other major barriers to enhancing hepatitis C treatment uptake. This is a care cascade from the United States, where only 50% of the estimated number who are living with hepatitis C are diagnosed. Obviously, small proportions that are disease staged and small proportions that have commenced treatment. Obviously, with the advent of interferon-free therapy, you would hope you could lift that treatment proportion very quickly, and the whole enthusiasm and optimism around these new therapies may also have an impact in terms of helping to drive forward increased screening and diagnosis as well. It's interesting to think about what we could do if we did have broadened access to hepatitis C therapy, but it would provide the foundation of HCV treatment as prevention. As Evan Wood has shown in the previous presentation and what's been able to be achieved in British Columbia with HIV therapy, we may be able to have the same impact with highly effective hepatitis C therapy. So we could think of several populations that you could evaluate hepatitis C treatment as prevention within. There's a community-based People Who Inject Drugs program that's moving forward in Melbourne, led by Margaret Hellard and Alex Thompson. We're concentrating on a couple of projects among prisoners and HIV-infected gay and bisexual men. I just want to briefly go over where we're up to with those projects. If we think of the settings of people who inject drugs, the unfortunate reality is that close to 10% of active injectors are incarcerated each year in Australia. And obviously that's replicated in many countries around the world. And we would like that to change, and we would like drug law reform to reduce that proportion, but that is the reality on the ground at the moment. We have about a third of our active injecting population who are on OST. So we believe if we can build infrastructures for rolling out interferon-free therapy in the prison and the OST setting, as well as having some community-based programs to supplement those two areas, that we may have an impact in terms of treatment as prevention. When I talk about treatment as prevention in the context of prison, some people say, well, there shouldn't be many hepatitis C new infections in prisons. But the reality is that they do occur. This is data from Andrew Lloyd, who's been leading a very large study looking at the incidence of hepatitis C in New South Wales prisons for over a decade. And it's about 10 to 15 per 100 person years incidence. So we designed, myself and Andrew Lloyd designed the STOP-C study with a primary objective to evaluate the impact of a rapid scale-up of interferon-free HCV treatment on HCV transmission. The primary endpoint will be the change in HCV incidence from the pre to the post scale-up phase. We have public-private partnership funding from Gilead Sciences and the National Health and Medical Research Council in Australia. We have a network of prisons selected. We've commenced the surveillance phase of the project where we're monitoring incidents for a 12-month period. We've started in Goulburn. We're going to commence in Lithgow in the next couple of weeks, and then we move into the other 3 prisons later this year. We've enrolled 300 inmates so far from Goulburn. We've a plan to overall 1,200 inmates into the surveillance phase. Our current chronic infection prevalence at baseline is 24%. The treatment scale-up phase will probably start in the second quarter of next year. The regimen will be sofosbuvir GS5816 for the obvious pan-genotypic potential. Obviously, this will require licensure, but we're assuming that things will go forward in terms of FDA approval and readiness for use of a licensed product. We'll be evaluating HCV incidence over a 5-year period. So it's a very exciting project moving forward, and obviously we'll be incredibly interested to see not just the outcomes, but just the feasibility of this sort of research in the prison context. It's a complex area to work in terms of research. There are clearly major issues in terms of research among inmates to deal with. If we move to HIV and hepatitis C co-infection in Australia, in fact we have a relatively small population who have co-infection. So our HIV population is just under 27,000, our chronic hepatitis C population is 230,000 estimated, but only about 10% of the HIV population have chronic hepatitis C. So we're looking at a 2,500 to 3,000 population. Given the relatively good cascade of care in the Australian context, obviously we need to get better, and we're trying to move towards 90-90-90, but even now it's not a bad sort of level in terms of the cascade of care. That provides the feasibility, we believe, in terms of treatment as prevention in co-infection. One of the key aspects is that a lot of the antiretroviral therapy in Australia is delivered by primary care prescribers at the primary care level. And that will be crucial in terms of upskilling those primary care prescribers to take on hepatitis C treatment to treat their co-infected patients. And then many of those primary care prescribers hopefully will also become prescribers for mono-infection populations, so we can expand the base of practitioners involved in hepatitis C broadly. So it's somewhat similar in a sense to STOP-C. Our primary objective is to evaluate the impact of a rapid scale-up of interferon-free treatment on transmission. Again, primary endpoint looking at change in HCV incidence. We've started enrolling into an observational database to characterise behaviourally clinically, we're doing fibre scans on everyone, and virologically the population. We have education programs we're rolling out, as I said, for HIV GP prescribers. We want to move forward with both broad and a targeted sort of hepatitis C treatment strategy, and those protocols are in development, and the surveillance systems are in place and being further enhanced to do the evaluation. What I think really underpins the feasibility of this type of treatment as prevention project is the recent recommendations by our Pharmaceutical Benefits Advisory Committee. So this is the committee that evaluates new therapies. So from the March 2015 meeting, the recommendations were that sofosbuvir and ledipasvir and sofosbuvir and declaraspvir be listed on our subsidised program without disease stage restriction and without drug use restriction. So we should get broad access in Australia to hepatitis C therapy very soon. It's very exciting. We may get that in December. There's a few minor tweakings within the price negotiations that are obviously ongoing, and it needs to be approved by our federal cabinet. But we're very confident that this will come forward. Just lastly, I'd like to make the obvious point to this audience that civil society has a crucial role to play in the development of a broadened access to interferon-free HCV therapy, and clearly that's been the case for HIV. I just wanted to highlight the HEP Coalition, which is a coalition of several community organisations and NGOs that are really being very active in this space at the moment in terms of trying to advocate and move forward for broadened access to interferon-free therapy. And there are many projects happening around the world that have been on the back of organisations that have been involved in HIV. And I'd really, I suppose, make a pitch to the HIV sector. We need the HIV sector to help move the agenda forward. This is one project through the International HIV/AIDS Alliance in Ukraine, where they've been involved in raising awareness, they've been involved in surveillance for hepatitis C within Ukraine, they've been involved in public health advocacy and trying to get the government to support the rollout of hepatitis C treatment. So it's very important that these organisations that have a background in HIV really lend their efforts as well to hepatitis C. Why should we push forward for interferon-free access for all? It's such an enormous advance of interferon-containing therapy. It really is revolutionary. Hepatitis C-specific quality of life impairment does occur in early liver disease, and it is reversible. So an argument to treat people across the disease spectrum. There's obvious potential HCV treatment as prevention benefits, although they need to be evaluated clearly. I think non-treatment equals ongoing monitoring potentially for decades. So to keep people engaged in that monitoring process may be very difficult. So I think often when patients are ready for treatment, it would be preferable to have treatment accessible for them. Maybe I'm too optimistic about this drug price curve, but I think the drug price curve will continue downwards in the coming years. And I think the ability to cure is empowering for the entire hepatitis C sector, and I think it really will move the field forward in the coming years. Finally, I'd just like to acknowledge a large number of contributors, both within the Kirby, Australian collaborators and international collaborators. Civil society have been very important in most of our projects, and clearly several funding agencies, both public and private. Thank you very much.